everyone, myself Roshan Kamwar, and today's topic is metabolism of fatty acid. In metabolism of fatty acids, there are two types of fatty acids metabolism. First is anabolism, and second is catabolism. Anabolism means synthesis of fatty acid, and catabolism means oxidation of fatty acid. In anabolism, we are going to discuss the synthesis of saturated fatty acid. Second is synthesis of unsaturated fatty acid and synthesis of branch chain fatty acid. In catabolism, in which oxidation of fatty acid occur at alpha carbon, so called as alpha oxidation, beta position, so called as beta oxidation, and at omega position or omega carbon, so called as omega oxidation. We first discuss about the anabolism of fatty acid, in which first anabolism of fatty acids occurs in different tissues, for example liver, kidney, adipose tissues, and lactating mammary gland. The enzymes for the fatty acid synthesis are present in these organs at cytosolic fraction of cell. So enzymes present in cytosolic fraction of liver, kidney, adipose tissues, and lactating mammary gland. It consists of three steps. First is synthesis of acetyl-CoA and NADPH plus H positive. Second is synthesis of malonyl-CoA from acetyl-CoA, and last is biosynthesis of fatty acid chain from acetyl-CoA and malonyl-CoA. For fatty acid synthesis, it requires number of substrate including acetyl-CoA, malonyl-CoA, and for synthesis there is requirement of fast complex. There is also requirement of malonyl NADPH plus H positive and ATP as energy source, and coenzyme A molecule, so etc. In first steps, synthesis of acetyl-CoA. As we know, when dietary carbohydrates and amino acids when consumed in excess can be converted to fatty acid and stored as triglycerides. So, in presence of high glucose concentration, amino acid concentration, this is converted into pyruvate, and that pyruvate transported to the mitochondrial matrix, where pyruvate is converted to acetyl CoA by the PDH complex and also converted into oxaloacetate by the enzyme acetyl CoA carboxylase. Now acetyl and oxaloacetate get condensed into the form of citrate by the enzyme citrate synthase, and then citrate is permeable for the mitochondrial membrane, so citrate transport to the cytosol, where it is converted to acetyl CoA and oxaloacetate by the enzyme citrate lyase. With the addition of coenzyme A, here acetyl CoA releases from the citrate molecule with the utilization of 1 ATP as an energy source. Oxaloacetate is converted into malate by the enzyme malate dehydrogenase. In this step, NADH plus H positive is act as a reducing coenzyme and it is converted to NAD positive. So with the production of malate, in next step, malate is converted to pyruvate. In this step, NADP is converted to NADPH plus H positive with the production of one CO2 molecule. So in this reaction, acetyl CoA and NADPH are produced. In second step of fatty acid synthesis, that is synthesis of malonyl CoA from acetyl CoA. Acetyl CoA get carboxylated by the enzyme acetyl CoA carboxylase. In this step, we required one ATP molecule as a energy source. So acetyl CoA is converted into malonyl CoA in presence of enzyme acetyl CoA carboxylase and biotin as a vitamin factor for carboxylation. Next is synthesis of fatty acid. In fatty acid synthesis, there are addition of acetyl CoA on ACP, that is acyl carrier protein of fast complex catalyzed by enzyme acetyl CoA ACP transacylase and get it converted into acetylase enzyme. In next step, acetyl molecule present on ACP is spontaneously transported to the SH group provided by cysteine. In second step, malonyl CoA transported on the cysteine sulfahydryl group provided by the ACP molecule, and reaction is catalyzed by malonyl ACP transacylase, so forming acyl malonyl ACP molecule. Acyl malonyl ACP molecule produced by releasing one SH coenzyme A from the malonyl CoA. In next step, beta keto acyl ACP synthase transport acetyl molecule from the SH group of cysteine and dissociation of CO2 of malonyl CoA, forming beta keto acyl ACP. In next reaction, beta keto acyl ACP reductase reduce the beta keto acyl ACP molecule by utilizing NADPH as a coenzyme, where keto group present on beta position get reduced into the hydroxyl form. So the beta hydroxyacyl ACP produce. In beta hydroxyacyl ACP, the next enzyme is dehydrated, which release water molecule from beta hydroxyacyl ACP and it converted into trans del 2 enoyl ACP. Enoyl ACP reductase enzyme responsible for the reduction of trans del 2 enoyl ACP. In this reaction, NADPH plus H positive is converted into NADP and the CH double bond CH is converted into CH2 single bond CH2, so forming acyl S enzyme. Now acyl S molecule transported to the SH group provided by cysteine in fast complex. After this reaction 2 to 6 repeated 6 more times, means reaction from malonyl ACP transacylase to the enoyl ACP reductase repeated 6 more times and finally forming palmitoyl ACP. On palmitoyl ACP, the last enzyme which catalyze reaction in which the palmitic acid released from the sulfahydryl group of ACP. Hence, palmitic acid, which is 16 carbon fatty acid, produced after this synthesis reaction. So, final molecule we get is palmitate, that is 16 carbon molecule. In fatty acid synthesis, if we want to synthesize a 16 carbon fatty acid chain, that is palmitate, we required 8 acetyl CoA, 7 ATP, 14 NADPH plus 14 hydrogen ion. Then it converted into palmitate plus 8 as coenzyme plus 7 ADP plus 7 PI plus 6 water production. In the regulation of fatty acid synthesis, the regulation occur at different steps. First is regulation on the most regulatory enzyme which catalyze rate limiting steps in fatty acid synthesis, that is acetyl CoA carboxylase. And second is hormonal regulation. In acetyl CoA carboxylase regulation, acetyl CoA carboxylase regulated by different mechanism. First, it is active in polymeric form and dephosphorylated form. Acetyl-CoA carboxylase inactive in monomeric form and phosphorylated form. The polymeric form and dephosphorylation induced by different mechanism. For example, the citrate molecule is responsible for polymerization of acetyl-CoA carboxylase, and insulin is responsible for dephosphorylation of acetyl-CoA carboxylase. The phosphorylated form is induced by glucagon signaling and epinephrine and norepinephrine signaling. The monomeric form is maintained by malonyl CoA and palmitoyl CoA. Acetyl-CoA carboxylase and fatty acid synthesis also regulated by high carbohydrate and low fat diet, while inhibited by high fat diet.